Hello everyone. Welcome to today's live broadcast, Expanding the Molecular Toolbox for Nanopore Sequencing. I'm Jackie Evans of Oxford Nanopore, and I'll be your moderator for today's event. Before we start, there's a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. If you want to go full screen, you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right, or use the Q&A button. We'll make sure we try to resolve any issues. With no further delays, I am pleased to introduce today's presenter, Dr. David Stoddard, Associate Director of Applications at Oxford Nanopore. David has worked with Nanopore technology since 2007, and his expertise has been instrumental in the development of the library preparation kits. He has a biochemistry degree from Wadham College, Oxford, and studied for his DPhil under Professor Hagan Bailey.
Hi, thanks for tuning in. So as Jackie says, I've had the Nanopore bug for a number of years now. My focus at the company is mainly on library preparation, so how we turn DNA and RNA into a form that is compatible with the Nanopore sensing device. I'm assuming that some people watching this presentation will know a fair amount about Oxford Nanopore's technology, and that some people won't know a thing. So I'm going to spend the first few minutes talking about the technology and how it works. This slide illustrates Oxford Nanopore's vision for a complete end-to-end solution from sample to analyzed results. The aim isn't to be just a company who makes a sequencer, but provide the whole end-to-end process from sample, be it blood, leaf, cell culture, whatever, through sample extraction, library preparation, sequencing, to full analysis of the results, so returning the biological information that the experiment set out to investigate. In terms of our sequencing devices, we have our PromethION, which is a scalable, high-throughput standalone device, which will be available very soon. In fact, you may have seen that we shipped our first one a couple of weeks ago. But for this presentation, I'll be focusing on the MINION, which is a lower throughput portable device, but uses the same sensing chemistry as the PromethION. So everything that I'm going to be talking about that's possible on a MINION will also be possible on the PromethION when you can get one of those. The concept of using nanopores to sequence DNA has been around for a few decades now. We can see a sketch here from the notebook of David Deamer, one of the godfathers of nanopore sequencing. In essence, you have a nanopore, a small hole sitting in a membrane of some sort. You have an ionic buffer on either side. When an electrical potential is applied, the current of ions flows from one side to the other through the pore. And as an intact DNA molecule passes through the nanopore, the current changes, the current signal being indicative of the sequence of the DNA molecules. And this is more or less how we do it today, as I'll explain. It looks simple, but the challenge is in making it robust and scalable and high throughput. So nanopores can sense any number of molecules. It doesn't have to be DNA or RNA. It can be protein and small molecules. But for this presentation, I will be focusing on use of nanopores to sequence DNA. Okay, so how does it work? You may have come across the use of solid-state nanopores in various contexts, and we have strong interests there, but the MinION device currently uses protein nanopores. And these have some distinct advantages for us right now, namely that a batch of protein nanopores will all more or less be identical to one another, and so behave in the same reproducible way. Also, we can modify them using conventional molecular biology techniques down to angstrom precision to enable us to tune the properties of the nanopores. Okay, so the nanopores sit in a membrane, not a lipid in our case, because this isn't robust enough, but it's a synthetic polymer which has high electrical resistance. We add a salty buffer solution to both sides of the membrane and apply an electrical current potential. The current given by the ions only flows through the pores, not across the membrane. And we measure the changes in the current as an analyte passes through. As an analyte passes through, the current is perturbed somewhat, and the current reading gives some information about the type of molecule that's passed through. And different molecules give different current deflections. So when this comes to being a molecule of DNA, the DNA molecule passes through intact, and the number of bases sit within the pore at any one time. So rather than sequencing a single base, you're actually looking at a small word. And so if, say, 5 bases fit into the pore, we're reading those 5 bases, and there are 4 to the power 5 different combinations possible, because there are 4 different types of base. So that's 1,024 different possible currents that you might see for any given word. And this sounds like a lot, but when we move the DNA through the pore 1 base at a time. So as a new word comes in to the pore, it's the same as the previous word, differing by just 1 base. So there are only 4 different possible transitions that you might have. And the current transition that you see will be dependent on the base that's just entered the pore, so G, T, A, or C. So over the course of an entire strand, we see a series of current levels, each of which is related to the previous one by 4 of the 5 bases. And we can use various types of signal processing analyses to convert this signal into base calls. So you see here the sort of yellow squiggle of different current levels as the strand passes through. So there are 3 essential components in nanopore sequencing. I've talked about the pore itself and the membrane in which the nanopore sits, but I want to talk a bit about the motor protein, which is the third important part of nanopore sequencing. So I talked in the previous slide about the need for the DNA to move 1 base at a time through the nanopore so that we can work out what the sequence is. If you just let the DNA go through the pore unhindered, it moves through far too fast, about 1 million bases a second, which is beyond our sampling frequency. So we just don't have enough time to see each base. So we slow down and control the rate of translocation of the DNA through the pore using a biological motor protein. And this controls and slows down the DNA and also acts as a ratchet, meaning that you see 1 base at a time, allowing us to measure the current levels. This motor protein is not attached to the pore. If it were attached to the pore, then if something happened to the motor protein, such as it denaturing, then you'd lose that pore for the rest of the sequencing run. So the motor is actually attached to the template strands via a DNA adapter, which is ligated on during the library preparation. As the strand is captured by the pore, the motor starts to move along the strand of DNA, feeding it through the pore 1 base at a time. The speed at which the sequencing occurs is dependent on the rate at which the motor protein processes the DNA. The motors we use are tunable, with the processing being controlled by the concentration of certain cofactors. Adjusting the cofactor concentration allows us to adjust the speed at will depending on the application. As well as ligating on a motor protein adapter, we also ligate on a hairpin adapter to the DNA. This covalently links the 2 strands in the duplex together. So during the sequencing, because of this hairpin, both strands of the duplex go through. So you sequence both strands at the locus. So as you sequence both strands, we call this a 2D read. And because you're getting 2 looks at the same position, the accuracy is higher compared to either strand alone, which we call, so either strand alone would be a 1D read. Once the template is finished translocating, the motor protein diffuses away and the pore is free to capture the next strand. We load templates into the flow cell at a concentration that is sufficient to minimize open pore time. So 1 strand is captured almost as soon as the previous one has finished. So the pores are sequencing pretty much all of the time. So this is a sort of closer look at the MINION. As I say, it's a handheld portable device, and this opens up the possibility of using it outside of a laboratory environment. The MINION connects to a laptop via USB, and the USB connection provides the power for the MINION, and it's also how the data gets transferred from the device. All the sequencing reactions take place on the flow cell, and that's the consumable part. The MINION is reusable, and the flow cell is the consumable. The flow cell contains the pores and the membranes, and it's where the library is loaded. Underneath the flow cell, we have a custom application-specific integrated circuit, or an ASIC, which is what controls the applied potential, and it's how the electrical currents are measured. The rest of the device is various bits of electronics and a fan to help control the temperature. Something I haven't covered yet is the sort of the real-time nature of the data generation. Strands pass through the nanopores intact, and the current levels are measured as this happens. It's measured in real time. As soon as the strand has started to pass through the pore, the data becomes available for analysis. This opens up a lot of possibilities, such as you can stop a sequencing run as soon as you have enough data to answer the question that you set out to. Or again, you can keep leaving the device running until you see what you want to see. It also means that you can perform something akin to sequence capture, except that you do it on device. So you sequence the first few bases of a strand, and if it's not from the region of interest or from an organism of interest, you can reject the strand, empty the pore, and search for a new one. This is called read until, and it's usually being pioneered by Dr. Matt Luce at the University of Nottingham, and he has already published some pretty neat stuff on this so far. So at any given time, 512 nanopores are capable of sequencing. Each nanopore acts independently and is individually electrically addressable. So all the signals are obtained from single molecules. And the single molecule nature of the sequencing means that we don't have any phasing issues, which can limit read length of some sequencing by synthesis platforms. Also, the electrical measurements used do not result in nanopore or motor damage, so that the quality of the DNA signal does not deteriorate over time. Therefore, there really are no limitations to the read length of the technology, and indeed it's been proven capable of producing very, very long reads. And as far as we can tell, the only thing which limits the read length is the quality of the input DNA itself. So if you're interested in having a go with the MINION, it is available, and I would encourage you to go to our website, www.nanoporetech.com, for more information about how to get hold of one. And you can also purchase consumables from our online shop. There's a strong and supportive online community where you're able to discuss ideas, get tips, and find collaborators. The participants with MINION pilot the technology, they develop applications and tools and publish on these. Also, we're rapidly developing products and upgrades and updates being released at regular intervals, and the community is the best place to keep up to date with the state of the art. So there have been plenty of publications, over 50, and they keep increasing in number. So publications and tools are being released from the Nanopore community over a range of applications, pathogen analysis, surveillance metagenomics, and tools and methods, including de novo sequencing and assembly. So all available through the community. Okay, so after the introduction, I'd like to move on to discuss what my work focuses on, which is library preparation. So library prep is the process of converting extracted DNA or RNA or amplicons into a format that is compatible with the sequencing system. Shown here for genomic DNA. So Oxford Nanopore's standard gDNA library prep is relatively quick and straightforward to perform, and it's capable of generating very long fragments of DNA, which will give you long reads. So to start with, the user can share their DNA if they like, but it's not a prerequisite. Obviously, if you want very long reads, then you wouldn't want to share your DNA, but it does make sense for some applications, and I'll come back to that. I'll come back to that in more detail in a minute. So after shearing or not, the fragments are then end repaired, and a single adenosine base is added to the 3' end. Then DT-tailed adapters are ligated on. One of the adapters is a hairpin, as we discussed previously, which connects the two strands in the duplex and allows for a strand to be sequenced followed by its reverse complement, all part of the same sequencing read. At the opposite end to the hairpin, we have a fork-shaped adapter, and this other adapter contains various features. It has a single-stranded section, meaning that the strands are more easily captured and threaded through the pore. It has the motor protein preloaded, which means that as the strands pass through, they go at the right speed. And it also has a region where we hybridize a tethering oligonucleotide. This tether has a hydrophobic group which helps drag all of the molecules in the library down onto the membrane, bringing them much closer in contact to the nanopores. And this increases our sensitivity several thousand-fold. After you've ligated on your adapters, we perform an enrichment step where we select for hairpin-containing molecules. This allows us to maximize the yield of 2D reads. So all the reads, so these are the reads where you observe both the template strand and its reverse complement. So the way you do this is after the ligation, you add an oligo which anneals to the hairpin, and this oligo also has a hook to facilitate subsequent binding to streptavidin-coated beads. So you add your tethered molecules to beads, anything with a hairpin binds, everything without a hairpin is washed away. And so when you elute the DNA from the beads, you are left with every molecule having a hairpin, so every read should be a 2D read. Now, we observe improved raw per read accuracy with the 2D prep compared to a 1D equivalent. As I say, because you get 2 looks at each strand, it's just slightly better. But recent improvements in our chemistry have led to improved accuracy with 1D reads. So now it's worthwhile as offering a 1D library prep protocol. And it works in a very similar manner to the 2D protocol. The main difference being that during the ligation, you omit the hairpin adapter and you don't perform a hairpin selection step. But no additional reagents are required in order to perform the 1D or the 2D library preparation. The kit is the same. So I mentioned briefly about users being able to share or not share their DNA. So just to return to that, unless you need very long reads, in other words, if you need reads that are greater than 20 kb in length, then we would normally recommend shearing. The reason for this is that it means that you're able to bring all of the fragments into a uniform range, which is below the sort of size where you may get unwanted shearing during the prep. So if you have fragments that are, say, 50 kb in length, this is the sort of size range where pipetting up and down could possibly shear them. So if you get shearing during the library prep, then it means that you won't have adapters on both ends of your DNA, which will mean that you'll get reduced 2D yields. So if you don't need anything, so if you're happy with under 10 kb reads, then we'd recommend shearing. The library prep is similar to other library prep protocols out there for other sequencing technologies, but 2 things to point out is that it is PCR-free. You don't need to PCR your samples, which is a laborious process, and it introduces bias, and you lose the epigenetic tags. Also, you don't need to do size selection isn't a prerequisite because there's no read length required. You don't need to do a size selection to remove anything too big or anything too small. You can just throw the lot at it at the nanopore and it will sequence what you give it. So it does have advantages over other library prep protocols. So when we launched the MINION in May 2014, we released a sample prep kit to go with it, and it was by far from perfect. So in those days, we did not perform a hairpin selection after the ligation, so there was no enrichment for 2D molecules. So you got a lot of 1D data, which you may not have wanted. As well as having a motor on the forked Y-shaped adapter, there was an additional hairpin motor to assist in the sequencing of complement molecules. This had to be loaded onto the hairpin at the very end of the prep. And early on, we made regular iterative improvements and upgrades to the protocol to generate improved yields, improved data quality, improve the customer experience. And we sort of made these regular improvements. And in August 2015, we released Kit 6, which included an upgraded motor protein, which allowed us to get rid of the troublesome hairpin motor. We upgraded the hairpin selection technique, which improved our yield. We increased the sequencing speed, which doubled the throughput. It represented a significant improvement in performance. And this protocol was and is performing very robustly in the field. We're now in the process of releasing our latest chemistry, Kit 7, and once again, this represents a significant upgrade in both sequencing chemistry, so improved raw read accuracy, and sequencing speed. We haven't just made improvements to our standard kit. We've released other kits during this time to expand the toolbox and empower customers to broaden their horizons. In September 2014, we released a cDNA kit, allowing customers to get sequencing data from their RNA samples. In June of 2015, we released our first iteration of a barcoding kit, allowing customers to run multiple samples in multiplex on a single flow cell. And in November 2015, we released a a kit facilitating sequencing of low input samples without the need for PCR. And in February 2016, we released a PCR-FREE barcoding kit. So as I say, we're about to move on to the release of Kit 7. And this is accompanied by a change in flow cell chemistry, namely an upgrade in PORE from R7 to R9. And we also changed the base calling algorithm from HMM to RNN, recurring neural networks. And these changes mean that our 2D accuracy, raw read accuracy, goes up to 95%, and our 1D raw read accuracy goes up to 90%. This 90% for 1D reads is higher than when we started for 2D reads back in May 2014. So as such, library preps that now offer 1D reads rather than 2D reads actually start to become useful. 1D libraries give you improved throughput over a 2D library as more time is spent looking at different fragments of DNA. In a 2D prep, it takes twice as long to sequence a piece of DNA as you have to look at both strands. So I'd like to spend a bit of time just looking at some of the new kits that we released and that we've been working on. So the standard kit requires an input of about 0.5 microgram to 1 microgram of starting material, and this provides sufficient DNA to reload a flow cell 4 times, or you could load 4 separate flow cells. If a customer had less than 500 nanograms of starting material, it was normally recommended that the template underwent PCR amplification to boost the level up to 1 microgram. But for certain applications, as previously mentioned, performing PCR is not necessarily appropriate. If you're concerned with bias, for example, if you're trying to assemble a genome which contains repetitive regions that may not amplify so well, then you might get reduced coverage in certain areas of the genome, which will hamper the assembly. Or if you're trying to identify different populations from a metagenomic soup, then you may not want to risk losing certain hard to amplify genomes that, for example, they may be GC rich and not amplify so well. It means that you wouldn't get a perfect, as good a representation in your subsequent sequencing of your starting population. Alternatively, you may be interested in looking at epigenetic tags in your template, you'll lose that information in the PCR. Performing PCR of long molecules can be a lengthy process, adding several hours to prep time. If you're concerned over sample prep time delaying your time to result, then having to perform a lengthy PCR is far from ideal. So having access to a PCR-free, low input prep would be attractive from several angles. And in order to fulfill this demand, we developed a PCR-free method of preparing a library in situations where you have a low input. And in fact, if you use this PCR-free low input expansion kit, you can, from as little as 20 nanograms of purified gDNA, you can obtain almost the same throughput as if you were preparing from 1 microgram of gDNA using the standard kit. And it's a very similar workflow to the ordinary kit. However, there is one subtle and significant difference, is that rather than eluting hairpin selected DNA from the strep beads, you leave the DNA on the bead and add the bead bound sample directly to the flow cell. The beads are heavy and fall towards the membrane in the pore by gravity, and this helps increase the potency of the library. In the plot lower right, you can see the difference between eluting DNA and leaving it on bead. So here we've prepped 40 nanograms of lambda DNA, and we either eluted the sample following the standard protocol, so off bead, or we followed the low input protocol and left the DNA on bead. And you can see that the on bead method generates about 3 times the throughput when you're talking about starting with low amounts. But I would like to mention that it's not to be a replacement for the standard library prep, because we have observed an as yet uncharacterized bias away from very long fragments when the library is left on the bead. In the experiment shown, we prepped a 20 kb amplicon, and the library was either left on bead, data shown in blue, or removed from the bead, shown in pink. And the upper plot shows the read length distribution observed for the 2 different methods, and you can see that when the library is eluted from the strep bead, you get a large proportion of your bases from the full length molecules, which is nice. However, when the sample is left on the bead, you observe an increase in shorter fragments. I mean, it's the same PCR product and it's the same beads, but there appears to be some uncharacterized phenomenon that results in a slight bias away from the very longest fragments when the DNA when DNA-loaded beads are added to the flow cell. In the example where the sample is G-tube fragmented, there's no significant difference in the read length. You can see from the green and the yellow plots that if you're talking about G-tube fragments centered around sort of 6 to 8 kb, then this bias isn't seen. Furthermore, the technique hasn't been validated for use with our wash kit yet, so we haven't tested the efficacy of the wash kit to remove beads from the flow cell. It may work, we just haven't tested it yet. So if I haven't done so already, I'd like to say that we haven't observed an upper limit to the read length of the MINION. The read length that you observe is directly dependent on the fragment length of the input material. So if you put short stuff in, you'll get short stuff out. If you have long fragments in your sample, then you should observe them. So here we show the image of complete 2D read that measured 255 kb in length. So this translates to 510 kb of DNA, pardon me, 255 kb of the first strand, and then you get to the hairpin, which connects it to its complement, and then you see 255 kb of its complement strand. The hairpin contains a sequence motif that is easily identifiable by the base calling software, and it indicates that the read is a bona fide 2D read. This sample in particular was from an E. coli prep where the DNA was extracted using a QIAGEN 500 tip. The read was aligned using BLAST, and it had a mean Q score of 10.1. As far as I'm aware, this is the longest complete 2D read that we have achieved to date. We may have achieved and sequenced some longer 1D molecules. I'm not sure about that. We are working on getting longer, but this will take improvements in our sample extraction and library prep technique, which is what is limiting our read length at the moment. So, as I've said, the read length that you get out of the MINION is a product of the fragments that you put in. Currently, we haven't found an extraction technique that does not produce some short fragments. And if you have any short fragments in your prep, even if they don't appear significant on a gel or a bioanalyzer, then you're likely to see them on the MINION. Even if the short fragments don't contribute much in mass to the final sample, they will have a high molarity compared with the long stuff. So, you know, if you've got 25 10 kb molecules, that'll have the same mass as one 250 kb molecule, but, you know, there's 25 more of your 10 kb molecules, you know, a large excess in terms of molarity. And if anyone knows of any extraction method that isolates or produces exclusively long fragments, then I'll be very interested in hearing about it. But in the meantime, until we've improved our technique or method, we've been working with the blue pippin from Sage to enrich for long fragments present in the sample. And we developed a library prep protocol to enrich for molecules that are over 40 kb in length. And the graphs displayed on the right-hand side, we take a sample of extracted gDNA, it's E. coli in this case, and we prep two different libraries. Neither are fragmented, but one is not size selected, that's the upper graph, and one is size selected, that's the lower graph. And you can see the striking enrichment for long fragments in the lower plot. It's not to say that you don't get the long fragments without the size selection, but they are accompanied by an excess of shorter molecules. The protocol, which should be released soon if it hasn't already, allows the user to achieve almost exclusively long fragments. The size selection is done as virtually the last step. It's done after the ligation of the adapters. We do it this way so as to minimize manipulation of the sample after the size selection. We've compared the size selection of input material, so you size select before your library prep, with size selection of the ligated library, and observe that you get better enrichment when the size selection is done at the end. We assume that during the library prep, you're getting some fragmentation during the manipulation, the pipetting, and the bead steps, and this is causing shearing of the DNA. So you're generating some small fragments during the prep. So if you do the size selection right at the very end, you minimize the manipulation required before running it. So even though, as I said, our standard library prep protocol is relatively simple, it takes about 70 minutes. It doesn't require the laborious process of size selection or amplification, but it still takes over an hour, even for a skilled user. And for certain applications, we envisage the need for a more rapid and a more simple library prep. for use in the clinic or in the field, where time to answer is critical or access to lab equipment is limiting. To fulfill this need, we've been working on a rapid sample prep and are in the process of releasing this kit and protocol, and it facilitates the generation of 1D molecules from extracted DNA in around 10 minutes. The kit is transposase based, relies on the fragmentation of template gDNA molecules and the simultaneous tagging of the fragmented template with adapter sequences using a transposase protein. Post transposition, the process of fragmentation takes about 2 minutes, and after the transposition, a sequencing adapter is ligated onto the adapted tagged ends and to facilitate the nanopore sequencing. Here we compare the output, the expected output of a rapid 1D kit with a standard 2D library, so a 70-minute sample prep time. In this case, the DNA again was using E. coli. So you can see that from the throughput graph on the right, in kilobases per channel per hour, the standard library prep does offer an improved throughput, but it does take an hour longer. And you also observe from the read length plots on the left-hand side of the graphs that you get a narrow range for the G-tube fragmented stuff. And the rapid prep offers some much longer reads, although the median fragment length does tend to be a bit shorter. So, you know, it does offer some opportunities for time to answers. It only takes 10 minutes. So I've called this slide decisions. As you can tell, we have various different library prep protocols and workflows and kits that on the face of things all seem to do similar things. You know, they facilitate the generation of sequencing data from extracted gDNA. But each protocol does have its own sort of advantages and limitations. So here I compare our various gDNA sample prep workflows and show the input requirements, the prep time, and the output that you might expect. I'm hoping this sort of comparison could act as a guide to help customers decide which workflow is for them, depending on what their sequencing priorities are, such as, you know, time to result, throughput, or read length. The highest throughput is achieved using the standard prep method, so G-tube fragmentation followed by adapter ligation. So if getting the most sequencing data is your aim, then that's the protocol you should use. The other methods gives different throughputs, and in part down to the nature of the different preps. So the rapid 1D kit, say it's transposase based, and when we run a transposase fragmented library versus a G-tube fragmented library, we observe a type of block not observed in a G-tube fragmented library. And we currently attribute this block to the transposase itself, and although it's rare, it does limit the throughput. The throughput of the unfragmented samples is reduced for a different reason. In this case, the molecules are much longer as they've not been fragmented, and therefore they're fewer in number. So their molar concentration is reduced, and it takes a bit longer for the pore to find a molecule to sequence. So the pore isn't sequencing all the time, and your throughput is reduced for this reason. Some very long reads also, so to talk about the read length now, if some very long reads are achieved with both the rapid kit and a fragmentation-free ligation method, but the majority of the bases sequenced are from fragments under 10 kb. So if you look at the graphs on the right, the sort of blue and the yellow plots, so there you've got rapid versus a non-size selected unfragmented. You can see that you've got some long fragments, but a lot of short stuff in there as well. If you want exclusively long fragments, then we recommend the size selection protocol, which is shown on the bottom right, because that gives you almost exclusively the long fragments. But alternatively, you know, maximizing your throughput or ultra long reads might not be what you need. You might need time to result satisfied very quickly. So maybe the rapid 1D prep would be for you. So I want to talk now a little bit about barcoding. Our barcoding kit allows users to pool and sequence multiple amplicons or small genomes in a single sequencing run, and this makes more efficient use of the sequencing power of the flow cell. In the first version of the kit, barcodes are introduced into libraries by PCR, so you attach generic adapters onto your templates by ligation. These adapters contain primer binding sites to facilitate downstream PCR. So the PCR is performed with a primer pair that recognizes the generic adapters, but also contain a unique barcode sequence as well. So the amplified products contain the unique sequence and are barcoded. So the different samples after they've been amplified with different barcodes. So then you take other samples and you can modify them with the same adapter, but amplify them with different primer pairs, and they'll have different barcodes on. So after amplification and cleanup, the samples can be pooled, and then sequencing adapters. So the sequencing adapters are the Y-shaped and the hairpin-shaped adapters. They can be ligated onto the pooled sample. All the samples are sequenced together, and the data can be separated out on the basis of barcode. It used to be that we just used to add a barcode onto one end of the molecules, but we found that we get an improved percentage of identifying barcodes when you introduce barcodes at both ends. So we now introduce barcodes at both ends, which offers us a substantial improvement. And we've recently expanded the set available from 12 barcodes to 96 barcodes. So to demonstrate the efficacy of this larger set of barcodes, we designed PCR primers to 96 different regions of E. coli, and we attached a different barcode to each amplicon. So following barcode deconvolution, each set of reads was aligned to the reference genome, and the coordinates of hits were plotted in Spotfire for each barcode. The size of the plotted circle represents the percentage of reads at each position. And the 96 barcodes segregated cleanly, with 90% of the reads having a barcode identified, and about 98.8% of the reads had the correct barcode. So we also offer a PCR-FREE. As of February 2016, we now offer a method of barcoding without the need for PCR, so PCR-FREE. As I've said, PCR is not always appropriate. You may not amplify your sequences efficiently and evenly, getting bias. PCR struggles with long fragments, so there'll be a limit to the read length that you could do as well. So a PCR-FREE method would be attractive, and we currently offer 12 unique PCR-FREE barcodes. So in this case, the sample is DA-tailed separately using standard techniques, and a unique DT-tailed barcode adapter is ligated on. Excess barcode is removed by a B cleanup, and the barcode modified samples go then pooled. Again, generic sequencing adapters, Y and hairpin shaped adapters, are ligated onto the pooled mix before sequencing as normal. The graph just shows a mix of the 12 barcodes all being identified and separated. So from the timeline that I showed earlier, you can be sure that we're not resting. We're endeavouring to broaden the scope of what is possible in library prep. This is just what we've done so far, but we're hoping to expand our rapid toolkit and all of our toolkits, and you'll be able to hear more about the latest developments at our upcoming conference, London Calling. Some of you, changing tack slightly from library prep done by users, I want to talk now about our Voltrax product. Some of you may have seen this. It's in development now and hopefully will be available to customers soon. For those of you who aren't familiar with the device, essentially it's a MINION-sized sample and library preparation device. It's fully automated with all the library preparation steps occurring on the device. It's entirely powered by USB, and it makes the whole process truly portable. And easy and reproducible as well. We envisage this, removing the need for complex sample prep procedures and expensive lab equipment, and removing user variation as being very attractive. At first, we hope to offer a standard ligation-based 1D and 2D sample prep and a barcoding workflow to allow the processing of multiple samples. We've run some preliminary experiments, and the results are promising at this stage. Eventually, the device will be compatible with all of our library preps that we have, including cDNA and PCR-based preps. In the first instance, customers will be adding extracted DNA to the device, but ultimately, we'll be tuning the device that is capable of extracting the DNA or RNA from your sample, as well as performing the library preparation. So the user will just have to add a bit of their sample to the device. Again, you'll be hearing more about this product at London Calling. So I'd like to finish now, and the time I've got left, with a few applications of the technology. Firstly, I'd like to talk about de novo assembly of microbial genomes. So using short read sequences to try and assemble a genome is like taking say a jigsaw, or rather, because you want 30x, 30 copies of the same jigsaw, only with each one cut slightly differently, and then all the pieces mixed together. The short reads will, the equivalent of having very small jigsaw pieces, and to find whether two pieces fit together requires a lot of searching. It makes for a substantial computational challenge, and the results tend to be imperfect. It's particularly difficult to assemble genomes that contain repetitive regions, for example. The bigger your pieces get, say 5 kb instead of 100 bases, then the task becomes a lot easier, and you can start to resolve repeats that are a few kb in length. As the read length goes up, the job gets even easier. So 50 kb, it gets much easier. We aren't at the point yet we can routinely generate reads of 500 kb, but as I say, we have seen, we've seen over hundreds of kb long. It's just really a question of getting the integrity of the DNA. So, if you imagine E. coli, which is about 4.5 megabases, if you've got 500,000 bases, then it's only 9 pieces of the genome. So the challenge becomes not less of a computational challenge and more trying to make sure that you can extract and manipulate DNA in such a way that they don't break. So we wanted to test our ability to assemble a genome using only nanopore reads. We started off with something small, and the DNA, we used the DNA kindly donated by Chantal Abergel from Marseille, and the DNA comes from quite an interesting source. They discovered a virus in Siberian permafrost that had been there for more than 30,000 years. The virus particle is enormous by viral standards. It's 1.5 microns long. And they were able to resurrect this viral particle in the lab. As they point out in their paper, as the permafrost is thawing out, the abilities of viruses to come back to life after tens of thousands of years could have some quite worrying ramifications there. Anyway, we weren't interested so much in the biology, we were more interested in assembling the genome. And as it turned out, it was fairly AT rich. So unfortunately, we had to PCR the library, which was unfortunate. But we didn't have much DNA, so we had to PCR it. And with a lot of help from Nick Lohman, well, to be honest, he did the whole thing actually, we were able to assemble the whole genome into a single contig. So that's one fragment covering the whole genome. And as you can see in the MUMMA plot of the assembly, against the published reference, things match up well. So the next slide is an improvement on this. Sometime last year, a landmark paper came out in Nature Methods by Nick Lohman, Josh Quick, and Jared Simpson. They demonstrated that it's possible, using only nanopore reads, to assemble a bacterial genome into a single contig. With very high percent identity, 99.9% identity eventually. So we wanted to see, that all sounded great, but we wanted to see if we could do it ourselves. So we took some E. coli DNA that we had, that we happened to have in our -80 freezer, and we made a couple of libraries. One of the libraries was a standard library they'd made by fragmenting the gDNA as described, and the fragment length distribution is around 6 kb. The other library was prepared in a similar way, but just omitting the fragmentation step, and it contained some very long fragments. You see from the pink plot, we got reads over 100 kb. And we wanted to see what the effect of having these long reads would have on the assembly. So we took 40x coverage of the 6 kb data. We put it through PBCR and Dismera correction and fed the reads into the Celera assembler. We got assembled into several, and this assembled into several contigs. When we repeated the assembly with a mixture of 6 kb fragments and some ultra long reads, so this time instead of using 40x coverage, we took 10x of 6 kb and 10x of longer fragments, and did the same thing, put it through PBCR and the Celera assembler. In this case, we got it to assemble into one contig, which is not all that surprising, but it is nice to see. So the bottom line is that long reads make de novo assembly easy. Just to hammer the point home, I do have one more slide on it. So for another project we had running, we isolated a flavobacterium from river water, which, as it turned out, was not in the RefSeq database. we assume it proved to be a difficult genome to assemble. It was found to be over 60% AT rich. So after a number of different sequencing runs, where some of the libraries had been site selected and some of the libraries hadn't been site selected, we had a large repository of sequencing data covering a wide range of read lengths, and we wanted to see what effect different read length libraries would have on ease of assembly. So we managed to assemble it into 3 contigs, one chromosome with about 5 megabases and a couple of plasmids. When we took 200 megabases, so about 40x coverage of 5 kb reads and above, that generated 15 contigs, so many more than were possible. But when we took 200 megs of 10 kb reads and above, that gave 3 contigs. So the same amount of data, just where the reads were twice as long, vastly improved the assembly. And the trend continues, and as you get longer reads, less data is required to achieve complete assembly. So if you take over 15 kb reads, that required 25x coverage. Over 20 kb reads required 20x coverage, and over 30 kb reads required only 15x coverage to assemble into 3 contigs. So if you've got long reads, you require less coverage to assemble. So even though site selection might seem like a costly route, it can make the final computational analysis easier and cheaper and ultimately better. I'll talk quickly about sequence capture. I'm running out of time, so I won't go into it in too much detail. Safe to say that large genomes, mammalian genomes and plant genomes, currently not currently possible on a single flow cell with a MINION. The output of a flow cell is in sort of in the gigabase range. It's well within the range of a PromethION, which is 300 times more powerful, but for a MINION, not yet. But to bring the biology of large genomes into reach, you can use sequence capture, where you enrich for the regions of interest using pull-down probes, which you can then, the regions of interest bind to the probes, which are specific for those regions of interest, and you can pull these probes out using beads, wash away anything that you don't want, and elute off the beads the region of interest, so you've enriched for it. And we tried this using, so what we did is we took the Agilent SureSelect small cancer panel, which is about 150 genes, and we got some sample DNA from Coriell, which contained a SNP in the BRCA1 region. We ran the standard protocol as recommended by the supplier, and we were able to detect the known SNPs in the target regions by doing sequence capture. We are planning to offer tools for detecting SNPs and other sequence capture workflows using our real-time data analysis software, and hopefully a protocol will be available. There are tools for doing this out, elsewhere, so you don't have to wait for that. Next, and finally, I guess I'd like to talk about a real-time application for species identification that we call "What's in my pot," or WIMP for short. As I mentioned earlier, as soon as a strand of DNA has passed through the pore, it can be base called and then analysed. In the WIMP workflow, species are identified and classified in real time. The classification relies, this classification, the output that you get is a taxon ID, for which a classification score is computed. The higher the score, the more confident that we can be that the classification was correct. In the output you see here, blue is very confident and yellow is less confident. And the user can set confidence thresholds, depending on whether the aim is to achieve maximum specificity or maximum sensitivity. This application is available through Oxford Nanopore. So the database was recently expanded to include viruses as well as bacteria. We wanted to test how well it worked, so we bought a range of viral RNAs from a commercial supplier. We prepared the libraries and sequenced them and put them through the workflow. So when we ran through the WIMP workflow, we obtained the taxonomic tree on the left. As I say, the blue was with high confidence and yellow was with low confidence. So you can see from the color codes that we were able to, the viruses were identified by WIMP with high confidence. As well as running the libraries through the WIMP workflow, we also barcoded each library so that we could look at how well the WIMP read counts corresponded to the actual reads we got. We wanted to know if the WIMP workflow would introduce any bias or if it was quantitative. So the bar chart on the right shows the two sets of reads agree closely. So the dark bar is for the WIMP reads, and you can see they match well with the number of reads obtained, number of barcode reads. So we were pleased that this was quantitative. This slide shows more data on the quantitative nature of WIMP, but as I'm running out of time, I won't go through it. I'll just proceed directly to a sort of worked example of WIMP. So we thought about looking at cow's milk. It's a good sample because drinking unpasteurized cow's milk is something of a fad at the moment, and although it apparently tastes amazing, there is a downside to it. The whole reason that milk gets pasteurized in the first place is that raw milk has got pathogenic bacteria in it. The table at the top shows that it's not uncommon for people to become ill from drinking raw cow's milk, and it's not massively surprising. The table below lists some of the pathogens commonly found in unpasteurized milk. Some pretty nasty stuff in there, Listeria and E. coli and so on. So we took some raw cow's milk and we left it on the bench for a few days at room temperature to let the stuff grow in it, and then extracted the DNA. And to make absolutely sure that we would have something to see, we spiked in some Listeria DNA into the cow's milk, and then we did the library prep and the sequencing and put it through the bacterial WIMP workflow. And when we analyse, we see lots of bacterial species present in the milk. They're ones that you'd expect to see, like Lactococcus and Pseudomonas. But what was quite interesting is that we saw a good number of species were identified with ones that are known to cause mastitis. Mastitis in dairy cows is particularly bad because it leads to necrosis of udder tissue, as shown in the photo. Now, the milk that we got was presumably from several cows, so we can't trace it back to the specific animals, but mastitis affects up to 50% of dairy cattle, so it's pretty certain that this was a real discovery of the disease, a real detection of the disease. And what we also see is that you can see the spiked in Listeria with very high confidence, and the number of read counts was more or less exactly the same as the amount that we added, as you'd expect from a quantitative workflow. And this kind of illustrates the sorts of things to which MINION and the associated workflows can be put, and highlights the non-lab applications and environments that you can use, that this can be used in. I mean, you can see how useful this workflow in combination with a portable sequencer would be in monitoring health and well-being of dairy animals. Our ambitions aren't limited to the confines of this planet. You might have seen in the press that there are plans to send a MINION up onto the International Space Station this year, making it the first DNA sequencer in space. Initially, the idea is to see whether or not it's possible to sequence in space samples that have been prepared on Earth, but in the longer term, the plan would be to use MINION to monitor astronaut health, monitoring intestinal microbiomes and water supplies, for example, but who knows, detecting extraterrestrial life maybe. So I will finish there. I just want to acknowledge all the staff at Oxford Nanopore Technologies, particularly in the applications department, Dan Turner and Dan Fordham, in the engineering department, Ant Jones, in our data analysis team, Fernando and Amber, and special thanks to Nick Lohman and Chantal Abigail as well. And with that, I'll finish, and if anyone has any questions, I'd be happy to answer them.
Thank you, David. The options available for nanopore sequencing are much clearer, as well as the decisions which need to be taken when planning an experiment or a new application. Before we get started with all your questions, here's a quick reminder for listeners about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get through as many as we can. Our first question today comes from Noel. How low can you go, David?
Oh, I assume that's referring to what's the lowest input that we can get. Right, okay. So I showed on a previous slide that we could achieve pretty decent throughput with as little as 20 nanograms input. We have gone down lower than this, and we have seen reads detected in the sort of tens of picogram range, so tens to maybe 100 picogram range. You do see reads, but at vast, at compromise of throughput, you do see a reduction in the throughput when you start with under 20 nanograms. But for certain applications, if you don't need to see many reads, if you're just doing species identification rather than assembly, and you're just interested in knowing the presence or absence of something, then you could start with lower than 20 nanograms, but just understand that you're not going to see the sorts of high throughputs that you might see if you started with 20 nanograms and above.
Okay, thank you for clarifying that. We have another question from Yayo. Do nanopore methods provide any advantage in sequencing bacterial genomes compared to others?
Well, I mean, the advantages of using nanopore sequencing, it's single molecule, it's PCR-free, and it's long read. So, the advantages that you'd have over sequencing I don't know if it's necessarily limited to bacterial, but yeah, the assembly is easier with the longer reads because it's PCR-free. You can see any epigenetic tags that you might be interested in that might be lost in PCR. It's within, if you wanted to do assembly, it's within range of the MINION as well. You can get good coverage, and do genomic assembly. I'm not sure if that necessarily answers your questions. I hope so.
Thank you very much. I think it does, along with the information that you provided in the presentation, a little bit like after the question came in. One more question that has come in from Scott is, can you identify epigenetic markers?
Yeah, that's a very good question. The advantage of not having to do PCR, and the fact is, you're not sequencing a copy of a copy of a copy. You're sequencing, the real thing. So yes, you can see epigenetic tags. At the moment, we don't offer data analysis software to deconvolute the epigenetic tags. But I believe that this is something that we're looking into and are in the process of trying to offer. But yes, you can see them. I think it takes a little bit of hacking at the user end to try and make use of the data. But it is in there.
Thank you very much. There's a question that's just come in from Daniela. And I'm going to abbreviate the question a little bit, but I hope that this is the key part of it. And it's regarding nicks in the DNA and whether they're actually the template DNA and whether they are a problem in sequencing.
Yeah, that's a good question. So if you have nicks sort of dotted around your template, then as the motor protein is processing the DNA and feeding it through the pore, if you get to a nick, then the strand will terminate there, the read will terminate there, you won't continue on, you won't get to the hairpin, and you won't go around the corner, you won't see the complement. So you would lose the 2D information because of the nick. You would still get a partial 1D read, be a bit shorter than the fragment length that you could have potentially had. But the 1D, because of the improvements in sequencing chemistry that we have, the 1D reads now are pretty good. So the data that you would get from that truncated 1D read would still be useful. But yeah, nicks would lead to premature termination and loss of data. They also lead to, or rather we believe that they lead to possible blocking of nanopores as well. We're not entirely sure how this works or exactly the mechanism for this, and I don't have a slide on it, but if you take genomic DNA and you treat it with an enzyme which will generate randomly gaps in your DNA, and you run it against a library that has not been treated in that way, you see a high degree of blocking and a much reduced throughput from the library that has been treated. You know, forgetting about the fact that the nicked and gappy sample also has a much lower percent of 2D reads and, you know, shorter read lengths, the amount of data that you get is also vastly reduced. So yeah, they are a problem in terms of the total yield, and the quality and read length of yield as well is also reduced. But you can try and you can remove nicks and gaps, or trying to minimize their effect by using a repair module from NEB called FFP repair, which has enzymes in it which should help ameliorate that problem, remove the nicks, resolve them, and seal any gaps. Alternatively, if you have a highly nicked sample, you can PCR it. The PCR fragments that you generate shouldn't have nicks or gaps in, and should perform better. You know, obviously you don't want to PCR, and you don't have to, but in this instance, you know, you could.
Thank you very much for that. I'm afraid we've run out of time for questions today. We will follow up for those of you that have posed questions via email, and you will also receive alerts from LabRoots for the on-demand supply of this session. So thank you for participating, and bye.